Good morning. I would first like to thank the scientific committee for the opportunity to present this work. Here are my disclosures. As we know, Intra-Host evolution represents the mechanism behind the continued emergence of new, highly divergent SARS-CoV-2 variant. It has been speculated that the emergence of antigenically diverse SARS-CoV-2 variant was driven by initially suboptimal immune responses and then was spread by forward transmissions. Persistent infection, even in not dramatically immunocompromised subjects, results in the continued local emergence of viral variants when associated with a high level of community transmission. The track of the emergence of viral mutations and SARS-CoV-2 variants by sequencing is an essential tool for public health. Therefore, aim of this work was to study the intra-host evolution of SARS-CoV-2 to assess conditions associated to the accumulation of new viral mutations. We analyzed 58 COVID-19 cases with 2 or more positive swabs collected longitudinally at intervals of at least 7 days from the first positive swab, as described in the graph. The study was conducted at the Luigi Sacco Hospital in Milan during 2021-2023 period. Only samples with a cycle threshold after real-time PCR minor or equal to 32 were sequenced by next-generation sequences with the Illumina platform, and only samples after whole genome sequencing with high coverage were considered then for a minority mutation analysis. Alignment, clade assignment, and mutation calling were performed using bioedit program, NestClade tools, and Stanford database, respectively, and association were evaluated with standard statistical methods. Table resume main characteristic of a study subject. The median age was 83 years, and the majority of subjects were male, 55.2%. All but one subject were hospitalized for a median of 23 days, and 12 died after a median of 25 days of hospitalization. No one reported a previous and documented SARS-CoV-2 infection. 79% were vaccinated, and 46.7% received 3 doses of vaccine. 7 subjects did not have any comorbidities and did not require any SARS-CoV-2 treatment, while 22.4% received more than one treatment. 60% of subjects with monotherapy received remdesivir, and 67.3% show at least 2 comorbidities. According to the study period, we observed several viral variants. 24% carried BA2 and 31% were XBB recombinants. Median days of negativization was 18 days, and we observed significant differences according to viral variants, with a longer period of negativization observed for BQ1 variant, 38.5 days, and shorter for BA1, 16.5 days. Swab positivity was observed after a median of 261 days of the last administration of SARS-CoV-2 vaccine, and no differences were observed stratified according to viral variant. A total of 906 single nucleotide variants were observed in all time points. The majority were non-synonymous mutation in 96.6%. The highest mean of non-synonymous variant density was observed in S gene in both T1 and T2 time points, specifically in receptor binding domain. Comparing T1 versus T2, 38% of subjects increase or decrease number of mutation, and 24% maintain the same number. By analyzing pattern of mutation, all subjects with the same number of mutation maintain exactly the same pattern, and 50% of acquisition and losses were confirmed. All new mutations observed at T2 were not present at T1 as minority mutations, and 50% of lost mutation at T2 did not persist as minority mutation. In the T2 versus T3 comparison, 67% and 33% increase or maintain the same number of mutations. respectively, and 75% of those with a constant number of mutation maintained exactly the same number by analyzing pattern of mutation, and 50% of acquisition were confirmed. 42% of subjects showed both acquisition and loss of mutation. Also in this case, all new mutations observed at T3 were not present at T2 as minority mutation. By analyzing the pattern of mutation in different genes, we globally observed a significant number of acquired mutation in both comparison T1 versus T2 and T2 versus T3. But while in the T1 versus T2, the acquisition were prevalent in S gene and losses in ORF1A, in the T2 versus T3, the opposite was observed. Stratified according to viral variant, in Omicron BA1, we observed a significant loss of mutation in all genes, while in XBB recombinants, the acquisition of mutations were prevalent in ORF1A, 1BAS genes compared to other genes. By analyzing the mutational pattern in different portions of main genes, we observed a significant number of acquired mutation, as expected, in S gene, in particular in the portion NTD and RBD. Subjects with cardiovascular disease showed a significant number of acquired mutation over time, and no differences were observed stratified according to different SARS-CoV-2 treatment or comparing monotherapy versus those who receive MOVE-1 treatment. Globally, we did not observe any mutation associated to drug resistance. In conclusion, although the exact biological mechanism of intra-host population dynamics remain to be explored, maybe in a large data set, our data suggests the mechanism of multiple steps in the fitness selection of SARS-CoV-2 mutation. The first step, corresponding to the first week, randomized mutations are generated, indicating a genetic diversity increase. In the second step, corresponding to the second week, purifying process was observed, determining reduction in the mutation number. A further increase of novel mutation was observed in subjects with prolonged infection at third week. The fixation of few mutations and the observation of undetected new one suggested a combination of selective pressure and diverse viral population, regardless of viral variant, and a limited role of minority mutations in predicting viral evolution. Globally, we observed that about 50% of all COVID-19 positive subjects develop a highly divergent viral subpopulation within 3 weeks of infection, regardless of treatment. I would like to thank all my colleagues that participated to this study and you for your attention. Thank you.